Well, hello, ladies. I hope you all are enjoying your nice break off school if you are someone who is getting that done now. If you are watching this on Monday night, well, I hope you enjoyed your break. So I'm going to try to keep this one pretty quick, pretty simple, because I do know that some of you all will be watching this over Easter break. But we're going to start chapter 17, and we're going to be talking about genes and variation. And your objectives are going to be: you will learn how evolution is defined in genetic terms, and you will also examine the source of genetic variation, which we'll see there happen to be three. So, just to give you a short review, we know that genes are carried on chromosomes, or our DNA, and heritable traits are controlled by these genes. And when we look at recent discoveries in genetics, they all fit perfectly with this idea and theory of evolution. Evolution makes all of this genetics make sense. So genetics allows scientists to discover how variation can occur, and they're also able to test and form new hypotheses about heritable variation in natural selection. So geneticists are able to look at the DNA, look at how some of these different variations occur, and they can kind of predict and see which way natural selection may be going depending on the environment. So the first thing we want to do is give you a little bit of genotype and phenotype, which hopefully you remember these words. If not, you can go back and look. But genotype is the particular combination of alleles an individual carries. And when we talk about phenotype, it's all physical, physiological, and behavioral characteristics. So the way we look, the way we act, that's phenotype. Phenotype is formed through the genotype and the environment. So when we talk about natural selection actually specifically works on the phenotype, not the genotype. Remember, it's the physical adaptations that determine survival with natural selection. So it's those physical characteristics that are being influenced, not the genotype. So some individuals are going to have a better phenotype suited to the environment that they are in, and these individuals we call are more fit. As we said in class last chapter, means they are more likely to pass on their genes and traits to the next generation. And remember, natural selection never, ever, 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 never acts directly on a gene. It is the entire organism that survives and reproduces, not a single gene. Okay, so I really want you all to think about that and know that natural selection occurs with the phenotype. So when we look at a population is a group of individuals of the same species that mate and produce offspring. So that's an important definition to remember. I would highlight that, circle that, do something with that definition. And because a population interbreeds, they're going to share a common group of genes, and we call that a gene pool. Okay? So a gene pool consists of all of the genes, including the different alleles that are present in a population. So a gene pool, I want you to think of a literal pool. And in that pool, we have all these different alleles for traits. For every single trait we have, there is an allele in that gene pool representative of it. And it's almost like picking out of those pools which alleles are present. So when we talk about allele frequency, it's the number of times an allele occurs in a gene pool. So I hope you all are ready for a little bit of math this chapter, because we will see some math. And this is compared to the total number of alleles in that pool for the same gene. So when we talk about allele frequency, frequency has nothing to do with whether an allele is dominant or recessive. It is strictly how often we see it. And in evolution in genetic terms, involves a change in the frequency of alleles in a population over time. So evolution, in terms of genetics, actually is looking at that overall gene pool number, and if we see a change in the frequency of alleles, then that means evolution is happening. Evolution is occurring. So this is why we say populations evolve and actually not individuals. So we can go back to that polar bear example. It was a population of bears that evolved. It was not just one individual bear that turned white that caused other bears to turn white and become polar bears. They evolved as a population and as a group. So here in this picture, we see an example of allele frequency. I'm not going to talk about this terribly too much because we really are going to focus on allele frequency in class and make it make more sense. But essentially, here we see a sample population of 48% heterozygous black, 16% homozygous black, and 36% homozygous brown. And you can actually calculate for each of these. For example, if there are 50 alleles, we can do math and see that there are going to be 20 black alleles, which are the black dots, or the big B's, and there are going to be 30 brown alleles, which are the little b's. We can do this for any type of trait, any type of gene we're looking at. We can look at the frequency of how often certain alleles occur. And remember, that's going to be the basis for how we build evolution in genetics. So when we talk about genetic variation, there's three sources that we're going to see: mutation, genetic recombination, and lateral gene transfer. So first we'll talk about mutation, which we've talked about before, but essentially it's a change in the genetic material of a cell. Many types of mutations. Some are a single gene. Some can be parts of a chromosome. Neutral mutations are not going to change the phenotype. Some mutations that can change a phenotype may or may not change fitness. It just depends on what the phenotype is and what we are looking at. Some mutations can be fatal, and some can even increase fitness in a species. So an example of increasing with a mutation would be the polar bears. A brown bear gave birth to a mutated white bear. That white bear had amazing survival, it had amazing fitness, and it passed on its successful traits to its offspring. So we do see mutations being both beneficial, neutral, and harmful. So every human is born with roughly 300 mutations that make us different and unique. Most are going to be neutral. We're not going to see it affect us at all. Some mutations we have may be harmful, but when we look at most mutations, they are going to be beneficial. And mutations only matter in evolution if they can be passed from generation to generation. So some mutations, like if you talk about altering your genes in a certain way, might not be able to pass on. So the only time a mutation really matters with evolution is when they can be passed on from generation to generation. So the second is genetic recombination and sexual reproduction, and this is where most differences between us and our relatives actually occur during genetic recombination. So this goes back to the idea of meiosis and that idea of crossing over. So if you don't remember that, make sure you look back at your notes. But each chromosome in a pair moves independently during meiosis. And when you add in 23 chromosomes and you have all of your genes, you get a possible 8.4 million different gene combinations with just your specific allele frequencies in your family. Now that's going to be a lot larger number when you start including everyone around the world. So again, crossing over, it's a way we see genes recombine. Crossing over occurs during meiosis and is when those paired chromosomes swap genetic information. So make sure you look back at your notes if you don't remember crossing over. It's in meiosis. I believe that was chapter 11. And crossing over increases genetic variability, period, end of story. So this is why we have things like where you can have your mom's eyes and your father's nose and your grandfather's hairline. There's so many endless combinations of gene possibilities with different combinations. That's why you can have a brother or a sister that looks so different from you and your parents, is because of all the different possibilities of genes. Lateral gene transfer is kind of a cool idea. It doesn't happen in us, but it does happen in bacteria. And you can kind of think of trading cards if you remember any kids in grade school with trading cards. But this is passing of genes from one individual to another that is not an offspring. So essentially, what can happen with bacteria is they can literally swap genetic information by passing each other and touching each other. And it can happen between the same species or different species. And this can actually increase the genetic variation for the species that gets the new gene. And this is important when we talk about this because of antibiotic resistant bacteria and evolution of single celled life. So when you talk about how all of these single cells originated and changed into all the diversity of life, it's because of lateral gene transfer, passing each other and passing different genes to each other. So it's like if you knew any guys that did trading cards in grade school, they would trade one card that they have for another card they really want from a friend. That's how lateral gene transfer works. Then we can talk about single gene traits, and these are traits controlled by only one gene. And single gene traits only have just 2 or 3 distinct phenotypes. So think of our widow's peak, our earlobes. Widow's peak, we can either have that nice V-shaped hairline or a straight hairline. We have earlobes that can either be attached or detached. So those are single gene traits. Polygenic traits, however, are traits controlled by 2 or more genes. And these are going to often have a lot more genotypes and even more phenotypes. So usually the phenotype is not clearly distinct from one another. And a great example of a polygenic trait is human height, because you always get into arguments with friends who's taller, and it's usually not by a lot. Well, that's why they're not clearly distinct. It's hard to tell them apart. So polygenetic traits will show what's called a normal distribution, which we can graph on what's called a bell chart. So this is a bell chart, and this is also called normal distribution. This is what, number one, teachers love to see on tests. Normally with that highest peak being around 85, 86. That's going to be your average. Then you have your lowest frequency of people up towards the very far right, and your lowest percentage of people who struggled on the far left. So if we put this in terms of height, I think right now the average is around 5'5" for female ladies, your height. So 5'5" would be at the very peak of that bell curve. And on the far right, you would have the tallest woman alive. And at the very far left, you would have the shortest, smallest woman alive, with everything else in between. So we'll look at bell curves tomorrow in class, and we'll kind of give you more examples and help explain this a little bit more if you need it. So I'm wrapping up now. So if you have any questions, comments, concerns, please write them down. Let me know. Really quick, what I want you to do is as soon as you're done reading this, there will be a video quiz. And what I want you to type in as the secret word, I see a box of Altoids here. So the secret word is going to be Altoids. Thank you and good night.